visual field. And I'm sure y'all slept on that over the weekend and got a grasp on how the visual field actually is projected and is thus reversed in each eye. Where the cornea and the lens focus the right part of the visual field on the left half of the retina of each eye, with the left half of the visual field focused on the right half of each retina. For the medial or nasal half retina of the left eye sees the same image as the lateral temporal half retina of the right eye. Therefore, the nasal half retina of the right eye will see the same image as temporal half retina of the left eye. But I'm sure y'all pondered on that and got that straightened out over the weekend. Oh, it's there in your book. It's there. It's already written. So accommodation comes down to focusing. And focusing brings us in to these different eye-related problems that we need to look at here today, primarily myopia and hyperopia. Astigmatism is an uneven curvature of the lens. We have a astigmatism chart. We're going to do it in lab today, y'all, or tomorrow. We're going to do 3, 6, 3, 7, 3, 8, and 4, 1 is the lab. It's a long lab, but those are the ones we're going to do, and there's a lot of procedures to do. So you'll get all of this lab here and part of the next one to work on it to get it done. And the astigmatism used to be difficult to correct because to correct it, you had to have an uneven curvature of the lens to fit the uneven curvature of your eye. Now it's one of those, I'll be done in an hour, come back, go shopping, come back in an hour, and we've got it. It's amazing what we can do with technology. Glaucoma is pressure in the eye, and too much pressure in the eye can actually cause damage to rods and cones, which can actually cause blindness. Surgery, and there's medications for that. The vitreous humor in the eye is constantly being made, and it constantly has an exit, so pressure is maintained about the same. If that exit or that small duct is ever blocked up, then that's going to build or cause glaucoma. That's not the only cause, but that could be one of the causes. So, age-related eyesight problems is presbyopia. Now, we talked about age-related hearing problems being presbyscus. Now it's presbyopia. Probably the most common age-related problem is just seeing things up close. We can still see afar, but seeing things up close, because to see things up close, the lens has to become more rounded to focus shorter or short vision. To see things afar, your lens is flat, and that's the way it likes to stay in an average day. So myopia is nearsightedness. And nearsightedness, myopia tends to, and this is kind of a more theory than realistic, but they'd say you'd have a long eyeball, and here's the lens, and when the light rays pass through the lens, they accommodate way before they get back here where they're supposed to on the fovea. So to correct myopia, we would use a concave lens which would diverge the light out here to make it come to focus way back here. So to correct myopia, we'd use a concave lens because it diverges light in front of the eye, spreads it further apart, so when it does come together, it's further back in the eye. Hyperopia is right the opposite of that. They would say you've got a short eyeball. When light passes through, it doesn't come together on the back of the eye, but beyond the back of the eye. It's real scattered at the back of the eye. So to correct a hyperopia problem, we'd use a convex lens. A convex lens converges the light sooner. Therefore, it connects in the back of the eye right here. Concave diverges the light out here. Convex converges the light in front of us. So that's how you correct. Of course, this one just an uneven curvature of the lens. And they talk about that on 291. 292, they have pictures of it. Has a doctor ever told you you had emetropia? Does anybody in here have emetropia? There was a time in my life that had told me I had emetropia. I said, well, how long do I have to live, Doc? Because I wouldn't have known what it was. But emetropia is good eyesight. Anybody not wear glasses have 20/20, 20/15 vision, very good. If you're close to 20, you still have hope for another 20 years. If you have good vision at 40, you might as well go ahead and schedule an appointment, because you're gonna need reading glasses. So it was about 42, 43 for me when I really needed reading glasses. And I need to wear them all the time now. I can sure tell when I don't. There's astigmatism. We're gonna talk about LASIK surgery a little bit. And then the next thing they get into are the rods and cones. Rods see shades of gray. They are extremely sensitive to light because they use a light-sensitive pigment called rhodopsin. Rhodopsin is made of a protein opsin and a derivative of vitamin A called retinol or retinine. Retinol or retinine, derivative of vitamin A, green leafy vegetables, everybody's favorite food, right? Go eat some spinach and you got it all covered. That's what Papa thought. You eat all kinds of green leafy vegetables, but usually anything green we steer away from. Y'all like green stuff? I love green stuff. Jed, no green stuff?
I'll eat it, but I don't like it.
I like spinach salad, like fresh.
In the salad? Okay, and and you'll see this as well. With age, your taste will change. You probably didn't used to eat it at all, but now you'll eat some of it.
Yeah.
And I don't know if that comes out that we're supplying our own food now, not our parents.
Maybe.
We appreciate a little more what we have in front of us now. Maybe that's part of it, I don't know. But without the derivative of vitamin A, rhodopsin cannot be built and you'd be night blind. So it's important that we do eat things that rabbits do. So in the presence of light, rhodopsin is destroyed. And in the absence of light, rhodopsin is built. In the presence of light, rhodopsin is destroyed because it's so sensitive, we wouldn't be able to stand light. It would be so sensitive. It takes about 20 minutes. 20 minutes for it to be built. You know, it breaks down faster than that, thank goodness, but if you've ever walked out of a dark room out into a bright sun, you can tell. I mean, it's cones. They're not sensitive because they work best with plenty of light, or they're probably not going to work at all. And we have a trichromatic color theory. We have red, blue, and green. And cones do use a pigment called photoopsins, and they do mention those in there, but they're not like rhodopsin when it comes to sensitivity. The red, blue, and green is your trichromatic color theory, and the combination of those three cones gives you all the variations of color that we tend to rename all the time. What used to be red may be fuchsia, maybe candy apple red. I mean, what's next? I don't know. But this tie-dye shirt illustrates, if you can see all the colors in that tie-dye shirt right there, there's some serious color over there. Purple, pink, green, blue, yellow, of course white. What else? Is there any other color? We don't have a cone for all those, but we see the purple because of the combinations of the cones. Now, a person that's partially colorblind typically will not be able to see two of the three. Anybody here colorblind? Which one can you see?
I can see them, but it's the borderline colors I can't differentiate.
Does red look brown to you?
Sometimes.
Yeah.
Yeah, it's red green is when I.
Yeah, and that, you can see, can you see a full-blown blue? Can you see his scrubs?
Yeah.
And that's typically the case. It's usually, seldom do you run across humans that are totally colorblind, like a lot of animals. I mean, the animals that see color are squirrels and fish and primates, our closest cousins. You know, those see colors, but dogs and cows don't see color, and I know y'all have this mystery in your mind, what about the bulls that chase the red capes? Well, Mythbusters fixed that for us a few years ago. Did you see it? That's pretty cool, wasn't it? And yeah, we stuck us a clown out there in a padded suit and a red suit, and he stood real still, didn't he? And then we had a red barrel out there, and it just sat there. Then we had wires running across it, and they'd zip these white flags and blue flags across on these wires. Guess what he chased? Movement. And when the clown went to moving, guess who got chased? In the red suit. Yeah, he barely made it to the barrel, where he got beat up in the barrel. But it was movement that they chased. It wasn't the red color at all. Now, even at full-blown color blindness, would definitely the tie-dye shirt would look brighter to you than the black sweater or the blue shirt, because of the light that's emitted. That's how people say, well, if you're gonna deer hunt and deer are colorblind, why do you wear orange? Keep getting shot. Because
Not for the deer.
people, it's people. You know, that's your biggest enemy. It's not the deer. But can the deer see the orange better than the camouflage? Absolutely, because it emits a bright array it emits light and its brightness. And sometimes they're not paying attention. If you're real still, they may walk right up on you and then just burst. Oh, what's that? You know, and they just see this. That shouldn't be in the middle of the forest, all this brightness. But there it is. And they know something's not right. Camouflage, yeah, they may pick you out, they might not, because it absorbs the light, see. So our trichromatic color theory is pretty cool, and there are lots of animals that see color. Birds is a big one that I didn't mention a while ago, that see color, but that's what we're based off of. And there's 5% of males and only 1% of females that may actually show some form of color blindness. So, and that's because you get your bad traits from your mother, right? And we learn that as well. All right, there's the dark adaptation, 20 minutes it says. Then it takes us over on 298, more of the trichromatic color theory. There's your rhodopsins and photoopsins, and there's your color blindness. They're actually saying men incidence as much as 8%, women almost 0.5%, so not even quite 1% in women. Now, we said the other day on the fovea, which is in the back of the eye, the fovea centralis is the area of most acute vision. Well, we said inside of it was 100% cones. Outside of it are your rods. So you actually see things at night better, not looking directly at it, but looking off to the side, using some peripheral vision, picking up the rods instead of the cones. It says on 299, there's 120 million rods and 6 million cones in each retina. This is why you see best during daylight. Only about 1.2 million axons enter the optic nerve. This gives an overall convergence ratio of photoreceptors on ganglion cells of about 105 to 1. But this is misleading because the degree of convergence is much lower for cones than for rods. In the fovea, for every cone you have a nerve. It's a 1-to-1 ratio. Will that not give you more precise vision? Yeah, it's not quite that good with rods, for sure. 1-to-1. Night vision is less distinct than day vision. You might see something over there but not make out detail. When there's plenty of light, we're making out colors and detail. And if we turned all the lights out and closed all the shades where we could see people in here, we could tell that Tracy has a more colorful shirt than other people, but could we tell much detail beyond that? We probably couldn't even tell that Curly's curly back there. That's how sad that would be. Neural pathways from the retina. This is on 299. And it's very similar to the pathway we started the class with. And it says the right half of the visual field is projected to the left half of the retina of both eyes. The temporal half of the left retina and the nasal half of the right retina. With the left half of the visual field is projected to the right half of the retina of both eyes, the temporal half of the left retina and the nasal half of the right retina see the same image. So something else to ponder on between now and Wednesday. Then we have, last here, we have the geniculostriate system and the tectal system. These are two systems that the eye feeds into, and they answer questions that we most often ask ourselves. Where is it and what is it? Which one of those questions should we actually answer first? Would you want to know what it is or where it is? It's hard to know what it is until you know where it is, where you get a look, isn't it? Well, 70 to 80% of the axons from the retina pass through the lateral geniculate bodies to the striate cortex, and this is a geniculostriate system. And it's involved in the perception of the visual field that answers the question, what is it? So we're more concerned, the mind is more concerned with what is it than where is it. To me, that's backwards. You gotta know where it is so you can look and see what it is. But maybe you can identify it once you, I don't know. Approximately 20 to 30% of the fibers goes through the superior colliculus into the tectal system, which answers the question, where is it? When I hear a noise, my first movement is to, where is it? And I may or may not recognize the noise. And maybe that's what they're thinking, is that we'll probably recognize the noise. And as we turn of where is it, we'll identify already what it is. I don't know. I need to know if I need to pull my gun or not. That's what I need to know. Real quick. So, neurocontrol of eye movements. We're gonna work on this, because you're gonna spin your partner in the chair tomorrow, if you have a partner. And hopefully they're not ease of stomach, where we don't mess up the floor. And we're gonna work with saccadic eye movements. Smooth pursuit movements would be following somebody walking across the street. It'd be something moving and you're following. That's smooth pursuit movements. Saccadic eye movements, have you ever seen something, and you guys hunt, I know you hunt, Jed. You ever seen something way out there and say, that's a deer? And you watch it, wait for it to move, and you watch it, and after a while it just kind of fades away. And you look off, and you look back and say, oh, it was a deer, it's gone now. But from a distance, your eyes will actually move to try to maintain the image and still bleach out over time. So we look off to get the image back, and we look back, and if it's there, you know, I don't know how many stumps I thought were turkeys over the years. Oh, there's one strutting in the field. It's a stump. So, but it happens all the time. We'll work on that. Smooth pursuit. Lateral geniculate nuclei, last thing in this chapter, on 302. The lateral geniculate nuclei says that the right lateral geniculate receives input from the right half of each retina corresponding to the left half of the visual field, where the left lateral geniculate receives input from the left half of each retina corresponding to the right half of the visual field. So that should have narrowed it down for you.
Who?
That cleared it right up, didn't it? I don't know, but when I read through the chapter and found those, I still don't understand them, but I like to read them. And I'm trying to simplify it the first time we talked about this. When we look at the brain in lab in a couple weeks, we'll look at the optic chiasma on the brain. And the optic chiasma, when I ask you on the test, what is it? You're going to say optic chiasma. Then I'm going to ask you, what does it do? You're going to say it's where vision crosses over. And that's what happens. But all this mumbo jumbo is basically saying part of what your right eye is seeing is going to the left side of the brain, and the other part staying on the right side of the brain, and same thing with your left eye. That's all it's saying. They've got their wicked ways of saying it. And I love to read it because the faster I read it, the better it is, you know.
I would spend 10 minutes reading that one thing.
I know, and still not get anything out of it, so that's one of those you sit around and ponder. And hope Mr. Dwann gives you a word of encouragement, because that sure did it. Endocrine system, chapter 11. This chapter, about everything we see in this chapter, we will see throughout chapters. Some chapters we may have already seen a little bit of it, but as we talk about the kidney, we'll see stuff talked about in here. Talk about in chapters 13 and 14, we talk about the heart. There's this stuff in here, so it's going to start overlapping. We we get to the reproductive chapter at the end, guess what, there's reproductive stuff in here. But they kinda go through it in a general, in this chapter, and then we detail it in the specialized areas. So, this will be more general with detail, but there'll be enough here that we'll be able to grasp where we're going. And there's one particular thing that we'll see in the kidney chapter, we'll see it in the heart chapter, and we'll see it in this chapter, and it's called the renin-angiotensin-aldosterone system. And when something like that's in 3 different chapters, do you think it's important? If it's involved in at least, it may be actually in 3 different systems, don't you think it probably involves every system in the body somehow, some way? So, that system you'll get to know quite well, because that's typically a essay question. You'll get to explain that back to me. And I'll try to clarify it where it's not as bad as the eyesight thing. So, give you some hope there again. I will say this. A guy went through this physiology class and went on the nursing program. He graduated nursing program. He's been out for several years, and he's worked in the, your dad would know him if I mentioned his name. He's worked in the ER, he's worked in intensive care, he's worked on the floor. He now is a dialysis nurse, and he likes it. He really likes the dialysis end of it. But this is how he got the job. And as soon as he got the job, he called me and told me this. Because this is about when I started heckling people over renin-angiotensin-aldosterone system. I don't know, it's just kind of a unique system, how everything kind of flows and falls together. So, he said he went in for the interview, and the lady that interviewed him said, "Why should we give you the job over anybody else?" And he said, "Because I know my kidney." "Well, what do you know that nobody else does?" "I don't know if they know it or not. This is what I know." And he rattled off the renin-angiotensin-aldosterone system just off the top of his head. And she hired him on the spot. She didn't interview anybody else. Because he talked about things she didn't even know, and she'd been working in there for 20 years. So, I mean, she probably knew it, but to spit it out like that, you know, that doesn't necessarily mean she understands things about it, but he just rattled. In fact, he's moved up that he's supervisor, and that he's gotta be there more often than a lot of them, but it's because of the system, so I blame myself for him getting a kidney job. But at the same time, there's a whole lot more out there to learn. So let's separate the endocrine glands from the exocrine glands. And earlier with epithelial cells, we talked about glands, and when we identified the fact that nearly all glands were made of epithelial cells except 2. And on 4.1 of this week's lab, it'll ask you what 2 glands are not made of epithelial cells. Can we remember those from our notes? We have them in our notes though, right? What kind of tissue makes the 2 glands not made of epithelial cells? Somebody's got to help me here. Y'all killed me. Their name gives them away. Which one?
The sweat or
Which pituitary? You got 2 choices. 50-50 shot. Come on. Shirley's withholding information back there. You have the posterior pituitary. What's the actual name of the posterior pituitary? Neurohypophysis. What kind of tissue is that? Nerve tissue, isn't it? Okay. The other one is the adrenal medulla. Is that not a brain name? Again, nerve tissue. Those are the 2 you're going to answer that question with. That was probably in your second day of notes, maybe the first day of notes that we did, first full day, somewhere in there. But that's what they're going to answer that with. So your definition between endocrine and exocrine glands. Endocrine glands secrete directly into the blood. Endocrine glands secrete directly into the blood, and exocrine glands secrete through ducts. That's D-U-C-T-S. They don't use ducts, D-U-C-K-S.
That makes me sad.
I got cracked up at my 2 little ones yesterday. I was putting out a deer feeder. And of course, as soon as I stopped to put out the deer feeder, they were looking for deer sign. Oh, my 6-year-old, I found a baby deer track. I don't know what it was when I got over there and looked at it. The gauge informed her it looked like a duck track. purpose glands. Dual purpose glands would be like the pancreas, the liver, the testes, the ovaries that fit both definitions. They may produce one secretion through a duct system and another secretion directly into the system, directly into the blood system. So let's look at this chart. We're gonna get through this chart as much as we can today, and then the reading kind of reinnervates the chart. Chart just kind of gives us a summary. Let's start with adipose tissue. Adipose tissue, we commonly know as fat, produces a hormone called leptin that decreases your appetite. Do you agree or disagree with that?
Seems like you would disagree with that.
You know, seems like the fatter I get, the more I still want to eat. In theory, if that does what it says, the more fat you have, the more leptin you produce and reduce eating, and you start losing weight, right? It should hold us an even kelter somewhere. Well, that's all bogus. Leptin does exist, but here's the kicker. Oh, this is difficult. For leptin to function, that fat cell has to be burned to produce leptin, which means you have to exercise. Did I say it? That hurt just to say exercise. Yeah, you have to exercise, burn the fat cell, leptin's produced, and then you're not as hungry. There's always you ever heard of leptopril or leptoprin advertised on TV? It's based off that theory. But they're selling the pill in form of which it's supposed to suppress your appetite without exercise. So they're saying you don't have to do anything but take this pill and lose. But now, don't buy it unless you're serious, because this is going to be weight loss. It's going to be 30 to 50 pounds. So if you're not serious about weight loss, don't buy this pill. I mean, that's what they're telling you. I don't know if it worked or not. It probably kills some people. Most of those kind of pills do. Beware of any of those weight loss pills. Weight loss is not as easy as taking a pill. It's more of habit. It's more creating good life habits than anything else in weight loss. So there, it does exist. It does suppress appetite, but you have to burn the fat cell to get the leptin. Zig, who exercises? I know, Jed, you exercise quite a bit. You do some running, and your wife probably still outruns you, but you still do some. There's no doubt about my wife. I just get behind her with a vehicle and shine lights so somebody don't steal her. I take her back home when she's done. I can't keep up with her anymore. But there's suppressing the appetite, the leptin, you have to burn those fat cells, and you have to create that habit more than anything else of burning those fat cells. It takes about 20 minutes to get to a fat cell, to get beyond your glucose reserve. So, who knows? Moving on to the adrenal cortex. Glucocorticoids and aldosterone. There's aldosterone. Glucocorticoids' primary target is liver and muscle. It influences glucose metabolism. So in times of stress, the adrenal cortex will produce glucocorticoids, which will give you more glucose. It gives you more energy to get through a time of stress. Makes sense, doesn't it? We need more energy to get through stressful times. Liver and muscles is where it's stored. If it's in the muscles, only be used in the muscles, then the liver can be used anywhere. Aldosterone acts upon the kidneys. It promotes sodium retention and potassium excretion. It's important that you remember that, because that's only a small portion of what we'll end up learning about the renin-angiotensin-aldosterone system. But if you can remember that, that's going to help in many ways. Sodium retention and potassium excretion. So aldosterone is going to be produced when you're thirsty and or dehydrated, because dehydration will kick in not only a thirst mechanism, but aldosterone, even another hormone that follows aldosterone, antidiuretic hormone. And by reabsorbing sodium, that will force our body to conserve more water. Everywhere salt goes, water follows. Remember that with the isotonic, hypertonic, hypotonics. So it's conserving that. Now, why excrete potassium? Well, it may not do it simultaneous to reabsorbing sodium, but it can if it needs to. But the reason we're excreting potassium, it has that ability, is we need an escape route for potassium. Potassium's one of those, if you don't have enough of it, it'll kill you. If you have too much of it, it'll kill you. So we need some way of getting rid, if you just go bananas one day and eat about 10 pounds of bananas, you might have a lot of potassium in the system. Now, does it matter if your heart stops in systole or diastole? No, the fact is your heart stopped. So too much potassium, yeah, we stop it at the top of its beat. Not enough potassium, we don't have enough energy to start the beat again, so we stop it at the bottom of the beat, at diastole. It doesn't matter, you're dead either way. So if you have too much potassium in the system, aldosterone can get rid of the excess. That's the good news of aldosterone. So there's only 2 times that aldosterone is going to be produced. When you're dehydrated, which you have low blood volume and low blood pressure with dehydration, or when you have too much potassium, that's when it's going to be produced. If everything's in the norm, we don't even need aldosterone. How often do you think that happens?
Not very.
We're just like a roller coaster. We're fluctuating all the time. So I would not advise you to eat a whole box of bananas at once. The texture would be too much for me. I couldn't squeeze down more than 1 or 2. All right, adrenal medulla, epinephrine. We all know that's your fight or flight. Heart, bronchioles, blood vessels, causes adrenergic stimulation. You may not understand adrenergic yet, but we will in chapter 9. Adrenergic is exciting. It's a speed up. Now I can do what I never could do before situation. So fight or flight. The heart produces atrial natriuretic hormone. This is a natural diuretic. This promotes sodium excretion in the urine. So we have ability to excrete salt. It's not a huge ability, but we have an ability to excrete salt. And this is one of them that's going to do it. And you'll see that listed in at least 3 chapters as well. Hypothalamus. Let me just tell you, if you're in doubt, just put hypothalamus. It's involved in a little bit of everything. You could arguably say it's involved somehow. So if it's a choice on a question and you're not exactly sure what the answer is, it's a good choice for you to put. It's got its finger in everything, whether it be your everyday mundane activities to excitement of thrival, of survival. It's there. It's involved. Releasing inhibiting hormones, acts upon or acts with, really, anterior pituitary, and regulates secretion of anterior pituitary hormones. Small intestine produces secretin and cholecystokinin, also known as CCK. Now, stomach, liver, and pancreas is your targets. It inhibits gastric motility and stimulates bile and pancreatic juice secretion. Now, secretin inhibits gastric motility because the small intestine is probably already full. So we slow down gastric motility so we don't dump too much in the small intestine. And then it promotes cholecystokinin to produce more bile so we can break down the stuff in the small intestine. So that they work together. And they also have found CCK, cholecystokinin, used as a neurotransmitter. Used as a neurotransmitter simply says that in the brain, it lays down information whether we like or dislike a food. Now that may change over time, obviously, but they find it here causing bile release to emulsify fats. They find it in the brain. You eat something, say, man, that's good. So the next time you see that food, you think, man, that was good. Of course, you could be like my niece that saw that food and says she tried it when she was 2, and now that when she looks at it, she said, I tried it when I was 2 and I didn't like it. She has a very closed mind about it, but maybe her taste buds have changed. And boy, if it ever does, I'm gonna give her a hard time. The poor kid would have starved to death in my house. All right, ready to move on to the islets of Langerhans, which is insulin and glucagon. These are 2 of the most antagonistic hormones in the system. And I like to ask antagonistic hormones, so that's most obvious ones out there. And there's a couple more I'll point out as we go. And one may be on this test and another may be on a later test. But insulin promotes glucose uptake. Glucagon puts glucose in the system. They're antagonistic, right the opposite. Kidneys, erythropoietin. We know this one from our first lab test, right? Kidneys see the blood the most. Kidneys determine if we need more blood, produces erythropoietin, goes to the bone marrow, produces more red blood cells. That's it. That's a pretty simple one. The liver produces somatomedins. We don't hear this much, but it's supposed to stimulate cell growth and cartilage. Well, cartilage is worse than watching plants grow. It is slow. It's slower than Christmas ever thought about being. I can't even explain it, it's so slow. Most of the time we lose and wear out cartilage, and I don't think anything ever grew back. So if I was gonna argue with one on this chart, it would be that one. Because you tear a little piece of cartilage, oh, we'll cut it out. You think it'll grow back? Not in your lifetime. I don't know what we do to improve it either. They say you can take the sharp cartilage and things like that, they'll improve it, but I have my theories, but none of them's good, so we'll move on. That's liver. Ovaries. Estrogen and progesterone, your two primary female hormones. Estrogen, of course, promotes female characteristics, secondary characteristics. Progesterone is the hormone for pregnancy. That's not the one that determines if you're pregnant, it's the one that maintains the pregnancy. The one that determines pregnancy is HCG, human chorionic gonadotropin. So those two are primary hormones of the female. Your parathyroid hormone increases calcium concentration in the blood. This is an antagonistic hormone to calcitonin. Calcitonin is the last one listed on the page for the thyroid. Parathyroid makes sure you have enough calcium in the bloodstream. Calcitonin makes sure parathyroid doesn't get carried away. The whole idea here is that we have enough in our diet, so these two don't have to get carried away either way. But if for some reason you're deficient in calcium intake, then we'll take some from your bone and make sure you have enough in the blood. If you don't have enough in the blood, it'll kill you. It'll stop your heart, it'll stop, muscles don't contract, it'll probably just stop breathing first, because your diaphragm not moving and you're not breathing, that'll stop the heart real quick shortly thereafter. So these are two good hormones to have. We actually discovered the parathyroids by accident. The parathyroids, we were actually researching thyroxine in dogs. We thought we had thyroxine figured out. We were gonna take out the thyroid gland in dogs and supplement thyroxine in their diet, and it was gonna be fine, right? Well, the dog died. So they drew blood and run tests and did the same thing to another dog, and the other dog died, drew blood, run tests, saw almost very similar, but almost exact results. And both of them were lacking calcium in the bloodstream. And it's like, the thyroid don't have anything to do with the calcium, but yet it did. Because the thyroid produces calcitonin, but it was the parathyroid glands that were kind of seated right in around the thyroid, side by side glands, and when it's taking the thyroid out, it's taking them out unknowingly. Now they know to leave them there. Because when they took those out, calcium metabolism messed up. When their dietary calcium dropped below normal, they died of asphyxiation. So now they know. It cost us a couple of pound dogs, but now we know. Pineal gland. Oh my gosh, we can talk about this for a long time. This is one back in the early 90s. They did tons of research over this gland. And this gland produces a substance called melatonin. And the things that we really know about it is that too much melatonin can actually cause depression. And they actually call it winter depression or sad disease. Sad, it would be depressed, wouldn't it? Seasonal affective disorder. So it's directly correlated with light. You ever hear of any of those diseases around here? I mean, we have depression, but is it because of melatonin? Usually not. So seasonal affective disorder, SAD, or winter depression, usually is found up north, northern United States, Canada, Alaska, when you have half the year in darkness or twilightness, there's not much light. What happens is lack of light increases melatonin. An increase in melatonin causes depression. So they go to the doctor all depressed. Do you think he gives them Zoloft? No, sticks them in a room with bright light. Go sit in there for an hour. Come back tomorrow and do it again. Come back the next day, do it again. you'll be fine. That's all it is. So we don't see it around here because we have this alternating daylight and dark every day of the year. And, I mean, with electricity, you see a whole lot less of it. These lights aren't the brightest lights, but they will play a role in melatonin production, so when it's dark outside, we go inside and flip the light up. Now, in the natural world, in birds, where one of the studies were done, they saw that as daylight increases, melatonin decreases, and the bird's gonad starts to grow to set up for reproduction in spring and summer. So they tried to apply that to humans and say, well, melatonin affects sexual reproduction in humans. We have so much artificial light that it really, I mean, it would play a role if we lived with chickens. Daylight, dark, it really probably would play a role, but it doesn't because we walk in, turn on the lights, and we just mess up the whole cycle. So we can prove this right here. How many of y'all were born in January? February, March, April, May, June, July, August, September, October, y'all are messing with me now. November, December, there was just about 3, maybe 4 months there somebody wasn't born in. When you get in a larger class, literally there's somebody born in every month, which says this is not influencing this at all. But I often wondered, having daughters at home, that if I put them in a dark closet long enough, they'd build enough melatonin that they wouldn't even like boys. I'm thinking about that with my little one, because she really likes boys. My wife says, you're gonna have to spend a lot of time with that girl. I think I'll strap her in a tree stand all winter with me, I think. Dear God. I'll sleep and she's strapped in, and I know she can't get in trouble that way. But there's your melatonin, and when we get to that chapter, there's a few other things I'll point out. They think it plays a role in onset of puberty as well. So when we get to that part of the chapter, we'll look at it again. Posterior and anterior pituitary, mark that spot right there, we're out of time. Now this is the anterior pituitary. We'll start there. There's quite a bit to say about all the different ones involved in there, and we'll start there when we come back on Wednesday. I'll see y'all laughing.